Let's go a little bit further into how the bacteria uses the embedded phage DNA from its own genome, looking at the DNA in a bacterium, Streptococcus pyogenes. In its genome, just before the CRISPR sequences, are called the CRISPR-associated genes, or CAS. Can you guess what their function is related to? These CAS genes are transcribed and translated through pyrogenase's intercellular machinery into proteins called CAS proteins that act as both helicases, which unzip the DNA strands like a zipper, and nucleases, which cut DNA like scissors. When CRISPR genes are transcribed into RNA, that is copied and turned into messenger molecules, an intermediate is produced, pre-CRISPR RNA, that turns into both CRISPR RNA and transactivating CRISPR RNA, also known as tracer RNA. CRISPR RNA holds a genetic code copied from the phage DNA in the CRISPR sequences, acting as the picture to identify the viral DNA. But it can't interact effectively with Cas protein on its own. It needs the help of its buddy, the transactivating CRISPR RNA, tracer RNA, to help bind to the Cas protein like a glue stick. Together, CRISPR RNA and tracer RNA form a guide RNA, or gRNA, that can bind to a class of Cas proteins called Cas9 to make a Cas9 gRNA complex. This complex can identify target DNA and break it. Let's take a closer look at the CRISPR-Cas genes in action. We find a virus trying to infect our S pyrogenase and has inserted its DNA into the bacteria. The bacteria notices and recognizes the DNA as one of a past infection, evident in the CRISPR genes. The bacteria's intercellular network signals for transcription of the CRISPR and Cas genes to try and fight off the infection. The Cas gene is transcribed and translated into Cas9 proteins, and the CRISPR gene, complementary to the section of the viral genome, is transcribed into pre-CRISPR RNA and processed into CRISPR RNA and tracer RNA, which becomes the guide RNA that fits into Cas9. Together, the Cas9 gRNA sequence locate a region on the viral DNA complementary to the CRISPR part of the sequence in the gRNA. Now you might be thinking, how does the complex know whether to cut viral DNA from its own genome, since both contain the same sequence? And you would be wrong to think that, since the complex doesn't have a mind and does not have the capacity to think. But you have the right idea, in that the bacteria does not want to have this complex cut its own DNA, because that would be an easy way to end the life cycle of the cell. What happens is the complex recognizes short sequences on the DNA just before the CRISPR-encoded regions, called the protospacer adjacent motifs, or PAMs. Without these PAMs, it is impossible for the Cas to cut. They recognize this. The bacteria's genome do not have the PAM sites this complex is looking for, so it will bind to and cut to the viral DNA, which do have PAM sites, and not the bacteria's. We'll talk more about PAMs later, but for now, this is what we've got after Cas9 opens the viral DNA. Notice how the DNA from the phage is complementary to the RNA originating from the bacteria's CRISPR system. What's happening at those sites that look like backwards Ds is that you've got a molecular kind of scissors that will break the DNA in these two places, creating a double-stranded break, or DSB. So if we make the cuts here, we get something like this with the viral DNA. This cut inactivates the DNA and renders it useless. The bacteria machinery cannot transcribe and translate this virus's DNA. Eventually, the viral DNA will unravel and disintegrate, with its parts diffusing in the cell. Thus, S pyrogenase is saved from the viral infection. Now what if I told you that we could hijack this system? Let's say we have a target gene that has a deleterious allele, or a version of the gene that generates problems for the cell. Say the cell is producing a machinery protein that weakly carries cellular cargo, like glucose, a major source of energy. We want to inactivate it, so we have an inactive deleterious allele in our S pyrogenase. making it stronger in this basic cellular function. We can do this by, instead of using the Cas proteins and the gRNA from the bacteria's genome, we edit the bacteria's genome using our own version of Cas9 and guide RNA. To do this, we engineer a Cas9 gene that can cut DNA without the PAM sites that were in the viral DNA. Then, we engineer CRISPR RNA to have our own sequence that targets the gene causing the deleterious allele. And finally, we fuse the tracer and CRISPR RNA together like a chimera to make a single guide RNA, or sgRNA. This way, it will be easier to control the experiment. In our cell, the sgRNA and Cas protein would bind together into a single complex. Together, the Cas sgRNA sequence locate a region on the bacteria's own genome, complementary to the CRISPR part of the sequence on the sgRNA. And remember, we've engineered our Cas protein to cut in the absence of PAM, so the bacteria's DNA will be cut by this complex. Cas makes double-stranded breaks here, and now we've inactivated this gene. What happens next is the bacteria will try to repair the gene in a process called non-homologous end joining. Pyrogenase will try to repair the gene by combining the two split ends on the DNA. When this happens, a mutation occurs where the gene isn't exactly how it was before. By inserting a new or deleting an old base pair in the gene now codes for a new protein that renders the effects of the deleterious allele mitigated. Thus, if all S pyrogenases has the deleterious allele you just removed, your new genetically engineered S pyrogenase has relatively more fitness than other S pyrogenase. That is, they are better suited to survive and produce more offspring without this deleterious allele holding them down. But what if, on top of removing genes that hold pyrogenase down, we were able to insert a new gene that raises them up? For example, what if pyrogenase could more efficiently metabolize the energy source lactose? Living just as well off less resources would be a massive win for pyrogenase on an evolutionary scale. We'll explore this in another video, but for now, I'll give you a taste. We can do this in a process called homology-directed repair, and you can use your own customized template of DNA to repair the damaged DNA with, effectively inserting your customized template into the bacteria's genome. Anyways, that's all I got for now. I'm the CRISPR guy. Take care now, and thank you for watching.